welcome everybody to our podcast LSTM in Conversation. So my name is Hilary Ranson. I'm a Professor of Medical Entomology here at the Liverpool School of Tropical Medicine. LSTM, we've just celebrated our 125th anniversary. We were established in 1898, and as such we are the oldest institute in the world dedicated to the study of tropical medicine. And over the past 125 years, we've really been delivering on our mission, which is to improve health outcomes for disadvantaged populations across the globe through research, education, and partnership. And during that time, we've had the opportunity to train some amazing global health leaders, and I'm absolutely delighted to be joined by one of those today. Before I start asking Yamni about her work on maggots, let me just say a few words of introduction as to her career pathway to date. Yamni graduated from King's College in London and came to Liverpool at that point to do a master's in applied parasitology and medical entomology. That took her to Swansea University for a PhD, a postdoc in Brazil, and then back to Swansea University, where she is now, and has established a research group on the use of maggots for all sorts of interesting purposes, which we'll hear more about today. Yamni is a very distinguished academic, having published over 75 different publications and book chapters, and is an advocate of public engagement in science. And I'm really keen to learn more about how you engage people and convince people that maggot therapy is really something that has a role in medicine today. So welcome, Yamni. Thank you very much for joining this podcast series.
Thank you very much indeed for inviting me on. Thank you.
So I guess I'd like to start really by just asking you, what is maggot therapy, and why should people be interested in it?
Okay, yes, brilliant first question. So the concept of maggot therapy is ancient, very ancient. We know that many civilizations and tribes and cultures had used maggots to help treat wounds that we now know in today's day and age weren't healing by themselves. They needed some intervention to kind of help them to heal. So modern day maggot therapy encompasses the very same principles that we have read about in the literature and in the world wars and battlefield surgeons who reported the presence, the infestation of natural maggots, maggot therapy, entering a wound, but those wounds were always reported to be non-pyrexic. There was no fever for the patient, the wounds were non-infected, and they were healing. So based on all of that, when we come to modern day times, maggot therapy is now a very clinical, takes a very clinical approach to this. So the maggots that we use are of a particularly non-invasive species, Lucilia sericata, the medicinal maggot, the green bottle flies maggots. And these are reared under sterile aseptic conditions, so sent out clinically to be used on patients to help them disinfect, debride, and heal their wounds.
So, as an entomologist myself, I'm keen to know why this particular fly. What is it about the maggots from the green bottle fly that make it particularly suitable for this purpose?
Yes, indeed. As you'll know, that maggots worldwide can be quite terrible and notorious for causing severe myitis, very, very severe infections and chronic problems all over the world, especially some of the tropical species that we have. However, the one species that we know is non-invasive and in particular cannot feed on healthy tissue. It will starve on healthy tissue. It's only able to feed on dead tissue. This is what makes Lucilia sericata, the green bottle maggot, absolutely ideal for the clinical use on wound care and wound management. Yeah.
So do you rear Lucilia at Swansea, and how do you do that? I presume you have to ensure that there's sterile conditions to produce these maggots. Can you tell us a little bit about that process?
Yes, of course. So we've reared maggots, Lucilla sericata at Swansea University, but we've reared them for research. So we didn't need to worry too much about any clinical output because we weren't sending them anywhere. However, there are factories all over the world which rear maggots clinically for use therapeutically. And the one that we have in the UK, and there's only one in the UK, and that's actually based in a place called Bridgend in South Wales. So very, about an hour away from my university, there's a factory called Biomond, who also have branches in Germany and I think in the States as well. And they are the clinical producers of larval therapy, or of larvae here. They send them all over the UK and all over Europe. But of course, they're living, tiny, sterilely reared maggots that are encased in a tiny little bag that are sent out to clinics and hospitals and surgeries all over Europe. But they will only survive a day in travel. So this is why the companies can't send them to greater, further away. But other companies that are in China and Australia and all over the world can provide them for their own countries.
So I think, how widespread are they in clinical use in the UK today?
Yeah, not as widespread as I would hope as an entomologist. As an entomologist, I came across and did my whole doctorate and postdoc on species that are pretty nasty to humans, vectors of terrible diseases. I mean, I looked at Chagas disease and I looked at sleeping sickness. And then when I came across to Swansea University and I got a lectureship here in anatomy, my head of school said, could you please create a module on wound healing for nurses? And it was then in 1997 that I discovered something called larval therapy as I was researching wounds. And I thought, well, what is this? And it transpired that only then, in the sort of late 80s, early 90s, had maggot therapy become a real thing in the UK. And it was mainly because we were seeing wounds that we couldn't treat. And you probably know this phenomenon of global antibiotic resistance, antimicrobial resistance, had started rearing its head in the 60s, but by the 80s and 90s, we were finding wounds that we couldn't heal. And so we went back to see, well, what was there that we were using to treat wounds that had infections and to treat wounds that weren't healing on their own. And it turned out that maggot therapy came up. So larval therapy was being reintroduced into the UK properly. It had already been around in America and Canada in the 1930s, in the pre-antibiotic era. They were actually using it clinically in the 30s. But then, of course, once antibiotics came on the scene, nobody wanted to use maggot. Why would you? When you can pop a pill for any scratch or itch or cough or whatever. And of course, we're now reaping the rewards of popping those antibiotic pills, and I think devastatingly so. So maggot therapy came into use. Now, it began with very few people using it, but the minute people started using it and they saw the effects, you can turn a wound on its head, which has been stagnant and non-healing for about 18 months, in 4 days with one treatment of 100 maggots in a small net bag. Well, they used to use free range, so they were loose maggots, but I think about 10 or 15 years ago, the company decided that they would put them in a little bag and the maggots could still do their job through that bag. So patients didn't have them crawling around, and nurses and healthcare professionals didn't need to worry about putting them on wounds. So the UK is using them, but not as widely as we would like. And we've been investigating all sorts of reasons as to why that is the case.
Yes, you have a wonderful video on your website, Love a Maggot, that sort of dispels some of the myths, I guess, and some of the concerns that presumably have arisen from your public engagement work about obstacles to rolling out the maggot therapy more widely. And I think one of the things I particularly liked from that was this issue about putting them in a bag so that people, I guess, maybe worried that they're going to start crawling all over them and spread, etc. And so I think that confinement, and then also I think concerns about will they emerge into flies was something else that's maybe you can talk a little bit about what are some of the reservations you've encountered when discussing this with patients and other groups.
Yeah, sure. So our work was carrying on. As a scientist work in labs, we were looking at funding and we were getting funding. And then in 2016, I got a rejection for a grant to look at the science of maggot therapy from the MRC. And I thought, well, why is this? And one reviewer had said, well, there is nobody that wants to use maggots, or that there's no, what's the point of doing this research when no one will want to use maggots? And I was really annoyed, not just at the rejection, but at that comment. And I thought, is that true? I didn't think it was. And of course, when I and my group started researching it, we realized that it was very true. And there is an inherent level of disgust at the idea of having maggots on you. A lot better now that they're confined, exactly as you say. But when they were free range and people would pour a little bit of saline on a little batch of maggots and pour them onto the wound and put them in a dressing, they would start, on occasions, if the dressing wasn't sealed properly on the wound, they could escape. And that would freak out a lot of patients, understandably. But maggots are in our radar as being tiny little disgusting varmints that frequent dustbins or on carcasses of dead animals, which all of these things make them ideal to eat the dead tissue off the wound, which is how they live, which is their survival. But the reservations are not just that it's a disgusting treatment, that people don't realize that it's the maggots that are used are clinically reared. They're under very, very strict sterile conditions, and the eggs are disinfected, and the little maggots that emerge are treated with such great care before they're packaged. So everything's done by the book clinically. But there are papers still coming out on it, and we ourselves have published a few papers on our research with the perception and the awareness and the perception of maggot therapy. And yes, it's still, even in 2024, we had publications saying 50% of patients and nurses find disgust, anxiety. They don't believe in the effectiveness because they don't know about it. There's disbelief that you're even using an ancient modality to try and treat modern day times. And so these are the issues that I thought we needed to address a little bit more. Hence, I set up the Swansea University Love a Maggot campaign to try and see if we could go out and talk to people about their fears, about their worries, and kind of make them understand that those are myths. The maggots that we are using, in 4 days, they will grow from the tiniest little emerging maggot onto the largest maggot, and at that point they need somewhere dry to pupate, and then they will take 2 weeks pretty much to pupate before they even get to the adult stage. So this idea that they will come off in flies, which is very much a real fear for a lot of people, can't happen. We remove the bag clinically after 4 days. I've worked with clinicians that have removed it after a week, and there's still maggots in there. They've not even turned into pupae. So that wouldn't happen. But these are the things we need to address. People genuinely feel that that's a concern.
It's really interesting, and I guess people do associate. I think my image when I first heard of this topic was sort of thinking back to war zones and people using it as a historical treatment. But of course, as you've said, with the increasing challenges faced by antimicrobial resistance and resistance to antibiotics, it's such a problem worldwide. This is alternative strategies are something that we all need to be thinking about. And so I'd like to ask you a little bit about the research side as well, because presumably the reason that these maggots are successful at healing wounds is they're releasing some sort of antimicrobial properties into the wound, and I believe that your team have looked into the properties of the secretions in some detail, and that's produced some very interesting findings.
Yes, actually, we now know that maggot therapy works in 3 ways on a wound. That the first way is to debride a wound, which is the most important thing that must be done before a wound will progress to heal. And debridement means getting rid of dead, necrotic, unwanted tissue, because that's a barrier to healing. It'll be in the way, and the wound bed won't be able to send good nutrients and good cells if you've got this barrier there. And not only is this barrier a nuisance to healing, but it's also a site for infection. It's something that bacteria would love to thrive on and grow on. So debridement is key, the first step to wound healing. Maggots feed on dead tissue. These species, Lucilla sericata, feed on dead tissue. So they're ideal to, within 4 days, get rid of that whole necrotic area that needs to be cleared. Now, we know how they do this. They do this by secreting enzymes. The enzymes, so they don't have teeth, they don't chomp, they don't bite. They literally secrete these enzymes. They can do it through the bag. The enzymes turn the wound, the tissue of the wound into a soup, which they can then drink up through the bag. The necrotic tissue is cleared. But if you think about where maggots live in nature, this species feeds on dead, decaying tissue, food, proteinaceous food, animals, carcasses, animals that have died in the wild. That's where you'll find Lucilia sericata swarming. I think it can smell death within 20 seconds. Some of the Reading researchers have shown that, they put little dead piglets out, and within 20 seconds, the first female Lucilia sericata had landed and laid her eggs. Those eggs will hatch and the maggots will start to feed. But if you think of what shares this feast with the maggots, we've got our primary decomposers, we've got bacteria, we've got fungi. So the Lucilia have learned to live alongside the bacteria and fungi by producing their own amazing repertoire of antibacterial factors. And the science over the last 2 decades has shown from all over the world that we have identified so many of these molecules. Our own group in Swansea have identified a tiny little molecule that we've called ceratocin, which is less than 500 daltons, and it's actually incredibly good at destroying resistant bacteria. MRSA was the main one that we worked with, but we've some researchers in the States took our molecule and showed that it inhibits bacterial cell division and causes lysis at greater concentrations. So, we know that the maggots have this ability to secrete in their sweat, in their spit, antibacterial factors, antimicrobial factors. And in fact, a German group, Anna Poppel's group, about 9 years ago showed that there are 47 distinct antibacterial factors produced by maggots, and the group actually have synthesized some of those and shown that they kill a range of bacteria. And remember, maggots are living creatures. They're evolving. They've been around, Diptera have been around for 200 million years, I think. So they've evolved to survive, and whether they've tweaked their antibacterial factors as they needed to, we think they're amazing, clever little things.
So I'm curious, and have you ever come across any microbial compositions in the wounds that proves toxic to the maggots? They can't disinfect, if you like.
Yeah, so we know that clinically there are a couple of species of bacteria that maggots struggle with. One of them, which is a major wound pathogen, is Pseudomonas, and Pseudomonas produces toxins itself when it's in the wound, and it's those toxins that can kill Lucilia. However, the Danish group of Anderson showed that if you double your dose of maggots, you can overcome the Pseudomonas infection. So it's just a question of using more maggots so that they don't succumb to the Pseudomonas infection. And again, a TVN recently contacted me and said that my patient had strep G, and strep G killed the maggots. So we are coming across a couple of bacteria which are, the maggots can't fight, and they can't. But one of the most interesting things that I will quickly say is about the inducible nature of maggot secretions. I don't know if you were going to ask me about that later. Sorry.
No, carry on, please. Yeah.
Sorry. Yeah, I just find it's just so much research has recently shown that if you have maggots that are surrounded by an infection, the antibacterial activity that they will now secrete goes up 6-fold. It's the inducible thing. Like if we had a cold or a virus that attacked us, our immune system would go crazy trying to destroy it. So maggots have that in them. They have this inducibility, which I think is absolutely brilliant, absolutely amazing.
Yeah, I was just thinking about sort of parallels to my work. I work on insecticide resistance in mosquitoes, and eventually, like antibiotics, same thing with insecticides, organisms evolve to develop resistance. And I just wondered if you or anyone else was exploiting this in the maggots and trying to actually select for Lucilia that would survive Pseudomonas or some of these other pathogens that are currently more problematic to cure with maggot therapy.
Yeah, no, that again would be absolutely brilliant. That'd be a great thing to do. We're not working on it as our group, but I believe there are groups that are looking at various ways to deal with these issues. But no, not in Swansea. We're not looking at that.
So you and others have identified a suite of antimicrobial compounds that are produced by these maggots. So do you think in the future there will be sort of recombinant productions of these, and they might be applied as a drug or some other form without the need for live maggots? Or do you think it's the benefit of the maggot therapy as you articulated that they clean the wound, they eat the dead tissue, and that really you need the live maggots to get the full benefits of these?
So as an entomologist, I am a huge fan of the living real thing. I think it's one of the very few insects that do a positive thing for us. We do have a lot of vectors and a lot of insects that are quite, as we know, quite devastating. But maggots are brilliant. They don't just debride. They don't just disinfect. We now know, again, the science has shown us that they produce a positive environment for wound healing. Not just a positive environment, positive molecules that are upregulated in the wound bed because of the presence of maggots, which aid healing. So the actual living maggot is doing so many things. It is a little factory of production of debridement enzymes, of disinfecting and antifungal molecules, and of promoting, accelerating wound healing. So that's great. That's the living maggot. However, we know that a lot of patients and a lot of nurses and doctors would prefer an ointment or a cream or something or a dressing that incorporates secretions that might work. Now that has been tried, and it's been tried I think in 2012 unsuccessfully. It's been tried a couple of times and it doesn't seem to work certainly as well as a living maggot. But I know, and don't know if I can say very much about it, but all of that is being looked at the moment. And I've got a PhD student that's very much looking at what we can do to harness some of these factors, remove them from the maggot and put them in some way onto a patient's wound without the need for the living maggot. Yeah, you're right. It's being looked at. And I think that is the ultimate aim for an awful lot of people. I still have reservations about it. I still think if you can tolerate the maggots, you're on to a winner.
Yes, because as you said earlier, I think that maggots are evolving creatures, and therefore you sort of evolve the therapy to adapt to the changing microbial
Yes.
composition of wounds and resistant bacteria as well. So I'm absolutely with you there that the value of having a living organism, if you can get over the cringe factor, it really is exciting as a and also a way of learning about. I guess that's a question really, is how much work are you doing or others in the space to really understand sort of the processes by which the maggot degrades the microbial composition of wounds, and are there things that we're interesting insights we're learning from just that process that might be exploited?
Yeah, there are several things going on in a wound. Quite often, you don't just have a planktonic bacterial infection. You've got a polymicrobial infection, which often leads to what we call a biofilm, formation of a bacterial biofilm. Wounds, over 80% of chronic wounds have a bacterial biofilm. And so we looked at, early on in our research, in about 2013, we looked at do maggots have any effect on biofilm? And we found that they did. We found that not only did they break up a biofilm once it's formed, so they've got a particular adhesion molecules that work to degrade a biofilm, but they also prevent a biofilm from forming. And that was amazing because the consultant microbiologist we were working with at the time said, well, he knows that one of the reasons why implants in patients and prosthetic tissues and so on fail is because they become infected, because they get this bacterial biofilm. So he went back to Germany and decided to coat or decided to work with surgeons who would coat their materials, their titanium or whatever, in maggot secretions before implanting, because we know that they prevent biofilm. So there are many processes involved. There's been key pathways that have been discovered for the healing processes by. In particular, the Chinese groups have discovered so much about how maggots are affecting various wound healing pathways and how their processes of disinfection occur and so on. So there's a lot. I mean, we know that we know how many antibiotics work in terms of whether they attack the cell wall or whether they attack cell division and so we're looking at all of the ways in which maggots do these things as well. And certainly, as I said, with ceratocin, we've shown that we get the bacteria unable to replicate. So they become, instead of dividing, they just become long cells, and which are no good at all in terms of replication and infection.
So does that have applications beyond actual wounds itself? Does that have applications as an antibiotic? So how generic is this effect against preventing cell division in pathogenic bacteria, I guess?
Yes, so of course we have focused all our work on looking at wound pathogens and wound bacteria because that's what maggots are licensed for wound care. But you're absolutely right. If you think about bacterial biofilm, for example, it develops in our teeth every night. The bacteria talk to each other and they develop this. We feel it when we wake up. This slimy. But the only way to get rid of it is to abrade it with a toothbrush. You could put maggot antibiofilm secretions in toothpaste. You could put it in, you could have a use for all sorts of things. One of the bacteria we tested in our laboratory was Mycobacterium, which causes TB, and Aceratin killed it, wiped it out. And so
Wow.
you think, well, okay, so yes, have you got a potential here for making this into a tablet, which you could use for TB, or even we found that it killed Clostridium difficile, which is a gram-negative anaerobic organism, which it was hard work to grow it up and work with it under anaerobic conditions, but it was wiped out by ceratocin, by this maggot secretion. So we know there's great potential for maggot secretions, and not just in the wound environment. And obviously you would then look to isolate those secretions, those molecules. You can't pop a maggot in your mouth and
I mean, that
it.
might take some persuading.
Yeah, indeed.
Can I take you back to how you first got interested in this? So you started off as an undergraduate biologist, and then what made you decide to come to Liverpool to study entomology and parasitology?
I was interested in insects since I was a small child, and I remember, much to my parents were quite horrified about it, but I loved insects. I also loved the human body. It was just something that I was fascinated by. So when I got to King's, I took every course in entomology that I could, every course in human structure and function that I could, and that reinforced my idea that I love both of those things. So Liverpool School of Tropical Medicine, and my interest was always in tropical medicine at that point. And how these diseases affected people, and malaria and other mosquito-borne diseases and so on affected people. So it was great to come to Liverpool and learn more. That course that I did, I think, taught me more than any other course that I've ever attended in my life. It was an awesome course, and it was literally what it says: applied entomology or medical entomology and applied parasitology, which was brilliant. So it was an eye-opener for me, and just inspired me so much to carry on my work in the field of insects and disease and human populations and so on.
That's fascinating. And while you were doing the course, I'm assuming there was an opportunity at that time to do a dissertation project. Did you do that on flies, or what did you do as your dissertation?
I did my master's. It was a research project, and I did it on. So we were allowed to work in the lab for 3 months after passing the exams in the summer. And my research project was working with a wonderful fellow called Professor Richard Ward, who was he was a medical entomologist, and he was working on sandflies and leishmaniasis. And he was working very much to try and control the vector. Lutzmya longipalpis was the one that we were working with, the species of sandfly that we were working with. So I spent 3 months in the lab with his group, looking at ways in which we could attract sandflies to traps and how we could stop them acting as vectors of this dreadful disease. And I loved working with him, and I loved working in the labs down in the basement at the Trop Shop,
Yes.
as it was commonly known then. And we published our very first paper. So after my master's, I got a PhD at Swansea University, working on other diseases and vectors of disease, but looking at the immunology of those insects. So I was trying to find out how they deal with the what is the effect of trypanosomes on regivid bugs, for example, or the effects of trypanosomes on tsetse flies. So that's how I became more interested in it. But the Liverpool School of Tropical Medicine just showed me so much inspiration. Richard Ward was awesome, and we published our first paper then when I got into Swansea, my first year at Swansea. So yeah, my first paper ever, and Richard was just a legend.
Yes, indeed. Yeah, it's interesting to hear about your recollections of that. I mean, I don't think the master's course is still running. It might have changed its name, tropical disease biology now, but it
Right.
really does follow a similar structure, and we still have the insectaries in the basement.
Yes.
Um, we also have some much let's say, more pleasant insectary conditions to work in as well as those, but they're still going strong. So
Excellent.
really interesting to hear about your time and how that exposure to the range of different vectors and vector-borne diseases has really sort of influenced your career since then. So you went to Swansea, as you said, and you studied trypanosomes, and then I think you to Brazil. Is that correct?
I did. We were very fortunate. We got a 4-month sort of, not an exchange so much. It was working with Fundaco Oswaldo Cruz in Rio, and they were looking at Chagas disease and Cruzi and Rangelli, and my work was there. So we went out there, and well, I went out there with my professor and at the time, and we worked together on looking at the various immune molecules that the vectors had against the parasites, to try and see if we could understand that a little bit more and try and work its way into trying to prevent the human condition. But of course, then after my postdoc, I was offered a lectureship at Swansea, slightly different path. They wanted me more to focus on human health, and then of course when I discovered this little insect, Lucilia sericata, it's like, oh, my world was back again. So yeah.
So these days, how do you split your time then? Because it sounds like public engagement is a large part of what you do, but you also run a research team, you teach. Tell us a little bit about what a day in the life of Yamni is like at Swansea.
It's crazy, I have to say. It's crazy. It's about juggling everything, isn't it? It's, excuse me, I'm sure you're all in the same boat. We do have a duty to our students, of course. You know, they need and they demand, of course, quite a good experience and a good learning experience. And so we try our best to provide that. So I teach on a range of anatomy and physiology and pathophysiology courses across to paramedics, to nurses, to healthcare scientists, and so on and so on. So that's great. And I love doing all of that. But then, of course, I teach the nurses, and the nurses they need to know about wound care and wound management, and they need to know about maggot therapy, because if I can get, and I've got it on the curriculum, if I can get them to understand it, then as they go out into the wards and into the world as nurses, they already have the background. That was one of our biggest problems. We found that nurses were not just reluctant, they didn't really have a solid awareness of it's an education and knowledge that needs really to be shared, I think, with healthcare professionals. But yeah, so the science side of things has become a little bit quieter. I collaborate now with other people that want to do the science, so they are working in our labs, and I, of course, I'm working with them, but I'm not in the lab myself all the time now, following, looking at the science. But there are other lecturers that I've encouraged to take on the mantle of maggot therapy and say, well, why don't you? And I know some lecturers look at honey and look at the effects of honey in wounds, so they are very willing to look at maggot therapy as well in that light. So the science is ongoing. The public engagement side has really gone mad. Because I think I'm very lucky, we are looking at maggots, and maggots evoke straight away all sorts of, oh my God, what is that? What are you doing? And so people want to know, which is great, because I want to tell them. So it just works out really, really well. So I've gone all over the place talking about maggots. I a little presentation, so it's not death by PowerPoint, but it always helps to talk a little bit with slides and images and so on. But it's more the interaction with the public, which is really great. So we have models. We built models of the fly, the larva, the pupa, so we can talk about the life cycle stages. We can talk about the action of larvae, and I always take along loads of maggots with me. So these are from the company, so they're clinical grade, sterile maggots, and we invite people to feel how maggots feel, to have a little hold of them and to have a look at them. We've set up stations where we have games, and the games are very much trying to get the maggot message out there, that if you've got a bunch of bacteria, and we've got tin can alley of bacteria in an infected wound, and you've got ping pong ball socks, which are maggots, and you chuck those maggots at those bacteria, you eliminate them from the wound. And that's the message that we try and get. And we offer prizes. We've got cuddly maggots, maggot mugs, we've got maggot key rings, and so on. So people take away a memory of what the maggot message as well. We've got leaflets and information and so on. So yeah.
So I guess you're taking it a sort of dual-pronged approach. You're working with the clinicians and trying to get increased understanding of the benefits of maggot therapy, and yet get that to be considered as an option with wounds, but obviously then also working with the broader public to sort of, if they do have a wound that might benefit from this, that they've got some understanding that can people request maggot therapy? How would it work if someone's got a wound and their clinical team and are not familiar with this? Take us through the sort of process.
Absolutely. So maggots went on prescription, NHS prescription in 2004, so they're available as a treatment. But you're absolutely right. The problem is not just a reluctance or a barrier to use because of anxiety. The problem is also budget and cost. So yes, anybody who knows about maggot therapy, and when I go out and do my talks, lots of people are sitting there thinking, oh my God, I didn't know this was a therapy. I didn't know that this would help in diabetic foot ulcers, or this would help my sacral ulcer or whatever. So they can request it. And unfortunately, it's still down to the clinician, the attending clinician, to say, yes, this wound is suitable. So first of all, assessment of the wound has to be done. But then they have to think, right, okay, I would like to order this. One tiny little maggot bag that will go on for 4 days costs £250. So it's an expensive treatment. So we're also up against it with cost. But again, if you think about 4-day treatment compared to a year and a half of maggot therapy, antibiotic therapy, or all sorts of other interventions, nursing time and so on, it's a winner. And cost-effective studies have been published to show that maggot therapy is way more cost-effective. But unfortunately, with the NHS in the state that it's in, it's all about the local budget. And I know that district and community nurses have to go to GPs to say, right, I think this wound is suitable for maggot therapy. Can I please order it? And GPs would look at it and say, well, no, because it's £250 and, we don't want to spend that. And so that's what we're up against as well. There are hassle factors of ordering and dispensing, which I think are also a problem. Was that your question? I'm sorry.
Yes, it is. No, that's really interesting. And it's making me think through the sort of practicality of it. So then, is it a home management treatment that you get your packet sent, or does it go to a GP, or does it go to home, and who puts it on, and then are you sort of discharged? Because I'm thinking about when you talked about cost effectiveness studies, I'm thinking about all the time potentially saved in clinical visits and follow-ups if it's something that can be managed in the home environment.
Absolutely, but it has to be clinically managed. So we know that obviously hospitals, it's fine because a patient is in treatment and needs a maggot therapy, and we've got TVNs and nurses around to monitor that. In the home environment, you can take a district and community nurse. She will order the maggots will arrive at the surgery. She will pick them up, take them to the patient, leave them, as long as there's an outer dressing on it. Sometimes if there's because the maggots tend to wash the wound out, so there is quite a bit of exudate on the outer dressing, and that can be changed very, very easily. But it's 4 days, 4 to 7 days. You can even leave it on 7 days. I know I work with podiatry in Morrison Hospital, and the patient can't come back for another week. So you leave the maggots on. They'll come in, they'll have the maggots put on, they'll go home. Obviously if the wound is on a heel, then you have to offload, so you'd give them a boot so they don't, and many reports have been of patients squashing their maggots accidentally treading on them. Absolutely. So that can very much work as an outpatient. People can have them, and they can have, you can have 2 or 3 applications, so the nurse will come along and say, well, it's almost completely clean and cleared, but I'm going to give you another 4 days. So you'd put on a fresh bag of L1 maggots, little baby maggots on that. So very much can be handled outside.
And generally for patients that have undergone this treatment, what's the perception been? I mean, you must have some success stories where persistent wounds have been cleared. I mean, are patients that have undergone maggot therapy good advocates for others, and do you use those as part of your engagement activities?
Absolutely, you're absolutely right. Once a patient has had a course of maggot therapy and has seen the results, quite often maggot therapy is still underused. It's a last resort treatment where those patients have had a chronic wound, a smelly, painful chronic wound for months. If you put on 4 days of maggots and they see that wound has changed and cleared, and their quality of life has suddenly changed as well, it's unbelievable. Because you'd be surprised. I met 2 patients in a car park as I was coming out, and we were talking about maggots, and they said, oh yeah, we were offered maggot therapy. We said no. And I thought, okay, why did you say no? And then there are those fears that, oh no, I couldn't stand it. And one of our surveys showed that whilst 91% of people would have maggot therapy if they were told they had to have an amputation, 9% still said no, they would rather have an amputation. And that stunned us, because that showed us
Wow.
that the fears are so real. People are petrified of maggots on their bodies for whatever reason. That they wouldn't. And so these are the fears we're trying to combat and talk about and so on. But you're absolutely right. Not just patients who see maggot therapy for the first time, our nurses who we know are like, oh, I don't want to do it. If they see the results, they're like, the TVNs are won over. They can't believe that you've got a therapy that works so well. And I have to tell you, there's a paper that's just come out of 3 case studies by an American group who had 3 patients that were life-threatening, multi-drug resistant bacteria in a sacral wound, a pressure ulcer on the back. And they had nothing else they could do. They put maggot therapy on all 3 of them, and they killed all the infection, and the patients survived life. And so you know that these maggots can do, really can combat life-threatening infections, and it's amazing. This was 2024 paper that just came out. So yeah,
it feels like all the things are lined up for this really to explode as in terms of its use in clinical practice. I mean, that, as you pointed out, there will always be some that are resistant to this treatment. But what, for you, what would success look like? What's your vision for this in the future?
Yeah, well, as an entomologist, we'd love it to be a mainstream therapy. It isn't a mainstream therapy. It's out there. It's certainly part of the armory of control of wound infection and for treatment of wounds, but it isn't used enough. Whether that's because of cost, whether that's because of hassles getting it. I mean, if a clinician orders a little bag of baby maggots, they've got a day. They're starving. They need to go on the wound. If the patient hasn't turned up or the nurse has gone off shift, you've lost £250, and that's part of it. The shelf life is part of it. I would love to see a slightly better way of delivery. Perhaps eggs could be sent and kept until they're ready to be put on the patient. Then they'd hatch out within the first 24 hours, and they would do that. I don't know. There's so many other ways. If we look across the world, people are looking at it. I know in Australia they're looking at sending drones with little bags of eggs to rural parts. In Africa, they're trying to use it. It's a cheap therapy. Obviously, in the Western world, it's not a cheap therapy because we have such sophisticated ways of trying to deal with things and sterilize them and so on and so on. But I know that many countries in Nigeria and Kenya, I use all these case studies in my talks, have shown very, very successful outcomes. So it's like it can be a worldwide therapy, but it just, it isn't there yet. That would be my dream.
Yeah, and I guess thinking about the cost, I mean, the cost of rearing insects with a complex life cycle, such as the Lucilia fly, that takes multiple weeks for generations to produce, and then doing that under sterile conditions. I guess it's not so much the case that it's expensive because demand is low. It's expensive because it really is expensive to produce flies under these conditions.
I think so. I think you're absolutely right. It is expensive to produce them. We know from our own rearing in the labs in Swansea that from egg to adult is about a month, and so it is quite a long process. But we managed to get our female flies laying on liver about 9 times. So we did find that sometimes we had so many eggs we didn't know what to deal with. How to deal with them. So and I know that the factory in South Wales in Bridgend, Biomond, they've got a very sophisticated process and a lovely way of manufacturing enough. That's not a problem for them at all. But you can see other countries maybe struggle a little bit with the cost of it. Yeah, for sure.
It makes me think of some of the mosquito control programs now that release of sterile males, for example, that require huge mosquito factories to produce these, and that does add expense.
Yes.
Of course, but nevertheless, I think in this era where one of the biggest threats to human health is antibiotic resistance, I think that the beauty of using a system that has evolved to feed and cleanse wounds using a living system to treat that is just beautiful, and I think it's really something that I imagine we're going to hear a lot more about in the coming years, thanks to the work of your group and others across the globe, and these sort of success stories that you, such as the case you just mentioned in the recent publication. I mean, that surely is going to be a stimulus for both demand from the clinical side and also from the patients, and let's hope that we see this becoming more of a mainstream therapy in the UK and in resource constrained settings where chronic wounds really a major issue. Thank you so much for your time today, Yamni. It's been fascinating hearing more about maggot therapy and about your own journey and your clear love of entomology. And I'm so pleased that your time at the Liverpool School of Tropical Medicine was sort of instrumental in your career journey. And we hope that we can continue to inspire the next generation of students and maybe we'll have more alumni from LSTM working on maggot therapy in the future.
Indeed, that's lovely. Thank you very much. Thanks, Hilary. Thank you.